The human story has always been intertwined with our quest to understand life itself. For millennia, we have selectively bred plants and animals, unknowingly manipulating their genetic makeup to suit our needs. Then, in the 20th century, came a revolution. The discovery of DNA, the very blueprint of life. This breakthrough ushered in a new era, one where we could potentially rewrite the code of existence itself, the era of genetic engineering. Early forays into gene therapy in the 1990s offered a glimmer of hope for treating genetic diseases, though these attempts were met with mixed success. However, the true turning point arrived with the advent of CRISPR-Cas9 in the 21st century. This revolutionary gene editing tool, often likened to molecular scissors, provided scientists with unprecedented precision and ease in manipulating DNA sequences. Imagine a world where we could eradicate genetic diseases like cystic fibrosis and Huntington's disease, effectively deleting them from our genetic heritage. Yet this power to reshape life itself comes with a profound responsibility, one that demands careful consideration of the ethical implications and potential consequences of tampering with the very essence of what it means to be human. At its core, genetic engineering is the process of altering an organism's genetic makeup, its DNA, to introduce new traits or modify existing ones. This can range from relatively simple modifications like making a plant resistant to a particular pest, to more complex endeavors, such as correcting genetic defects in humans. Imagine DNA as a long, winding instruction manual for building and operating an organism. Each gene within this manual is a specific set of instructions that dictates a particular trait, from eye color to susceptibility to certain diseases. Genetic engineering allows scientists to target specific genes within this vast library of information, either to delete harmful instructions, insert new ones, or simply tweak existing ones. The elegance of CRISPR lies in its simplicity and precision. It functions like a pair of molecular scissors guided by a programmable RNA molecule that can be designed to target specific DNA sequences. This revolutionary technology has democratized genetic engineering, making it faster, cheaper, and more accessible than ever before. The applications of genetic engineering are as diverse as life itself, spanning across medicine, agriculture, and even environmental science. In medicine, the potential of genetic engineering to treat and even cure genetic diseases is nothing short of transformative. Imagine a world without cystic fibrosis, sickle cell anemia, or Huntington's disease, diseases that have plagued humanity for generations. With gene editing tools like CRISPR, this vision is rapidly approaching reality. Beyond treating inherited conditions, genetic engineering also holds immense promise for tackling infectious diseases and developing novel therapies. Scientists are exploring ways to engineer immune cells to target and destroy cancer cells with pinpoint accuracy, potentially revolutionizing cancer treatment. Genetically modified mosquitoes are being deployed to combat malaria and Zika virus transmission, offering a novel approach to vector-borne disease control. Imagine crops engineered to withstand drought, resist pests, and require fewer fertilizers, promising a future of food security even in the face of climate change. While the use of genetically modified organisms, GMOs in agriculture, has been met with some controversy, the potential benefits for global food security and sustainable agriculture are undeniable. Section 4. A World Without Disease. The potential benefits of genetic engineering. The allure of a disease-free world, a world where genetic diseases are relegated to the annals of history, is a powerful motivator driving research in genetic engineering. The potential to eradicate inherited disorders like cystic fibrosis, Huntington's disease, and sickle cell anemia, diseases that have caused untold suffering for generations, is a tantalizing prospect. Imagine a future where parents no longer have to face the agonizing uncertainty of passing on a debilitating genetic condition to their children. Beyond treating and preventing diseases, genetic engineering also holds the potential to enhance human capabilities and push the boundaries of human potential. Imagine athletes with enhanced muscle mass and endurance, soldiers resistant to fatigue and injury, or individuals with improved cognitive abilities and memory. While these possibilities raise ethical concerns about designer babies and genetic enhancement, the potential benefits for individuals and society as a whole are undeniable. Imagine crops engineered to thrive in harsh environments, resistant to pests and diseases, and requiring minimal water and fertilizers. The potential to create a more sustainable and equitable food system, one that can feed a growing global population, is a compelling argument in favor of responsible genetic engineering in agriculture. Section 5. The Pandora's Box. Dilemma: ethical concerns and genetic engineering. The power to reshape life itself, while exhilarating, comes with a profound responsibility, one that demands careful consideration of the ethical implications and potential consequences. The ability to edit the human germline, making changes that would be passed down to future generations, raises profound ethical questions about designer babies, eugenics, and the very definition of human nature. Imagine a world where parents could choose their child's physical characteristics, intelligence, and even personality traits. The potential for genetic discrimination is another significant concern. Imagine a world where insurance companies deny coverage based on genetic predispositions, or employers discriminate against individuals with certain genetic profiles. Furthermore, the long-term consequences of genetic engineering are largely unknown. Introducing genetically modified organisms into the environment could have unforeseen ecological impacts, disrupting ecosystems and potentially leading to unintended consequences. Section 6. Drawing the line, navigating the global regulatory maze. The rapid pace of advancements in genetic engineering has outpaced the development of comprehensive regulations, creating a complex and often contradictory global landscape. Different countries have adopted varying stances on genetic engineering, ranging from permissive to highly restrictive. Some nations, like the United States, have adopted a relatively liberal approach to regulating genetically modified crops, while others, like many European countries, have taken a more cautious stance. The lack of international consensus on the ethical and safety standards for genetic engineering poses significant challenges for research development and the global trade of genetically modified products. The ethical considerations surrounding human germline editing are particularly contentious. The lack of a unified global framework for regulating human genetic engineering raises concerns about medical tourism, where individuals might travel to countries with more permissive laws to access gene editing technologies. Establishing a robust and adaptable global regulatory framework for genetic engineering is crucial to harnessing its potential while mitigating the risks and addressing ethical concerns. Section 7. The Future Unwritten. Genetic engineering in the 21st century and beyond. The field of genetic engineering is evolving at an unprecedented pace, with new discoveries and technological breakthroughs emerging constantly. CRISPR-Cas9, while revolutionary, is just the tip of the iceberg. Scientists are already developing more precise and efficient gene editing tools like CRISPR 2.0, which can target and modify DNA with even greater accuracy, minimizing the risk of off-target effects. Beyond gene editing, the burgeoning field of synthetic biology holds immense promise for the future of genetic engineering. Synthetic biologists aim to design and build novel biological systems, essentially creating life from scratch. Imagine microorganisms engineered to produce biofuels, biodegradable plastics, or even life-saving drugs on demand. As we venture further into the uncharted territory of genetic engineering, we are faced with profound questions about the future of humanity. The choices we make today will determine the course of human evolution for generations to come. Please subscribe, leave a comment, and ring the bell to stay updated with our latest content.